Hello, everybody. Welcome to this webinar. Good afternoon from Stockholm. Good morning if you're in the US, and good evening if you're in Australia. This is one of many webinars we've had in the Ebba Biotech series. My name is Ben from Ebba, and we're a Swedish company started in 2015 based on very interdisciplinary science, interdisciplinary collaborations. Before we get into the webinar, I just want to check that you are all okay, that you can hear us well, and that you can actually ask questions, because it's really important with these live events that we get some kind of interaction, that you can really ask the burning questions to the experts who are presenting. So if you just drop me a little note in the chat, let me know that you can hear me okay, and let me know where you are calling in from. It'd be really interesting to see. So the chat button is just at the bottom of the screen. Just click and just drop a note in there. I'll take a look and see who is messaging. But then just to continue about Ebba and our products, they are octotracers. So these are fluorescent tracer molecules that can label macromolecular components. And today we are going to be talking about Carbotrace. But it's not me that's going to be talking about Carbotrace. I am very excited today to introduce to you Dr. Tharagan Kumar. He has a background in biotechnology on the nanoscale, which led him to seek the link between science and technology and medicine. And he's focused heavily in his career so far on how modern technology can improve lives, specifically with making it more accessible. He recently graduated his PhD from KTH, the Royal Institute of Technology, and since then he's moved from academia into industry, and is now the development engineer at Isolate Medical, where he heads up the diagnostic platform, R&D. With that, it is my pleasure to stop talking right now and hand over to Tharagan. Welcome. Thank you very much.
Thank you, Ben. Thank you so much for the nice introduction. Thank you, first of all, for the opportunity, Ebba Biotech, because it's my pleasure not only talking about my own research, but how truly Carbotrace has helped my research in pushing it further. I'm very glad to present this webinar. Just quickly, thank you for the introduction once again. So I'm Tharagan Kumar, working as a development engineer in a company in Isolate, here located in Solna, Stockholm, Sweden. We are primarily focusing on cancer diagnostics, using liquid biopsy, but then we also have other products. If you're interested, it's called GeneMate. It's a hereditary cancer risk. If you're really interested, just check out that we have the service available. So just to get into the topic itself, I'm going to discuss about how cellulose is utilized in cancer diagnostic platform using microfluidic technologies. And in that part, how and where Carbotrace has helped me in this process. So this is my PhD topic, one of the papers that I published during my PhD studies. And just before I get into my topic itself, this study was done under Professor Aman Rissom, who's the head of the division, clinical microfluidics and point of care diagnostics. So it's a nano biotechnology division at KTH. So this research was done under his supervision. So I'm going to talk about, just give an introduction to the topic, a basic background about the cancer and cancer diagnostics, and some introduction to microfluidics and there are different types of microfluidics. And specifically in this presentation, I'm going to talk about affinity-based separation, and then cellulose in cancer diagnostics, how it is utilized, and finally give you a conclusion. So to get into the introduction, talking about cancer in a global perspective, it's the second leading cause of death, as we all are aware of, and then 1 in 6 person dies who gets cancer, and 70% in low and middle income countries are having a huge impact of this. And then only 26% of that even has pathology services to confirm whether it is a cancer or not. So it's really tough in remote areas. And it's a huge burden on our economy. It's approximately 1.6 trillion US dollars, and that's a huge burden, and that delays not only in just getting the treatment at the right time, but even the diagnosis at the right time. So it's a huge burden for us, for the economy. So talking about cancer, what exactly is a cancer? It's a disease that arises from normal cell due to genetic instability, and then it transforms into a tumor cell. It's not, that is where not the problem lies, but at certain stage, the tumor cell breaks from its normal tissue and then releases into our bloodstream and then moves to different parts of body, and that is where that is what is called metastasis. And here we can see a detailed view on how the vascularization happens, and the cells can be transferred from one location to multiple locations. So from one primary tumor site, it can affect different organs in our body, and this is where it's extremely risky for the patient when it reaches this stage. So the existing diagnostic methods are PET-CT MRI, very classical diagnostic method, and tissue biopsy, reaching out to the organ and then taking a small sample of tissue from the location and then checking for specific markers. But then the tissue biopsy is extremely painful in many cases and sometimes not even accessible. For example, in pancreas, it's not easily accessible to that pancreas location, and it's painful for the patient to go through. And PET-CT and MRI is usually expensive to handle these diagnostics, but that is what is used currently in the market as a golden standard. So how do we replace it? That is where liquid biopsy comes in. So what is liquid biopsy? In simple words, we just take a tube of blood and we will try to capture the cancer cell that is circulating in our bloodstream, so that it's extremely minimally invasive, and it doesn't hurt the patient as it does when it comes to tissue biopsy. So the idea is that one tube of blood will contain at least one or two circulating tumor cells, and that is what is our target. But also it contains many more information than the cells. That is, the tumor DNA that sheds from the circulating tumor cells can also be circulating in there. RNA can be there, and exosomes is also a very hot topic recently, which is being picked up by many research groups across the world, and it carries very vital information that we can identify what sort of cancer we are looking into. But in today's work, we will be focusing on circulating tumor cells specifically. So there are multiple enrichment technologies available in the market right now for the CTC, that is based on biophysical property, that is purely based on size, density, surface charge, or even deformability using microfluidics. That is based on, that is biophysical property. But when it comes to biological property, we purely rely on the surface markers expressed on the surface of the cell. In this case, for example, EpCAM is a classical example that is expressed on the surface of the cell, and we target those markers using a anti-EpCAM antibody, and then we can modify the microfluidic surface or a magnetic particle, as you can see in the first example of magnetic separation. Or in the last example, as what I've mentioned, is silanize. So basically, we modify the microfluidic chip with certain chemicals, and then we add antibodies that are against the markers expressed on the cancer cell, and then we flow the blood, and then we will hopefully capture the cancer cell. And that is what I'm going to focus on today. Instead of silanizing, we use a specific technology to modify the surface, and I will get into detail why is it better compared to other techniques. So I will quickly give you an introduction to the microfluidics itself. What is microfluidics? So as the name itself very clearly says, it's a micro, it's in the micron level, and then we can manipulate the fluid flowing across these microfluidic chips. So that gives us the ability to manipulate whatever is within the fluid. So that is basically what this microfluidics is. This microfluidics is used in different fields. I'm just giving a few examples that is used in our previous lab. That is, for example, cancer cells, spheroids, and even for SARS-CoV-2, we had some really good, we utilized the microfluidics platform in a very good way. And then HIV diagnostics, sepsis diagnostics was also a very good example of this, considering the size of the bacteria and microbioreactors. 3D printing using these different biomaterials and micro nanoplastics. That is a very interesting project also that is currently it is going on in our team, previous team. So these are a few examples that how microfluidics has been exploited in different fields. And today we will talk about cancer cell, how we have been using it for. Now coming to the project itself, that how cellulose is used in cancer diagnostics. So this is a published article in a scientific journal. So it's the topic is multi-layer assembly of cellulose nanofibrils in a microfluidic device for the selective capture and, most importantly, the release of viable tumor cells from whole blood. That is very important. So from the title, we can clearly see that it's a multi-layer approach of building multiple layers inside a microfluidic chip. And then the most important part is we can selectively capture only the CTC of interest, and then also we can release it for further downstream analysis, which is very important. What is the overall aim of this project is basically that when a patient has been diagnosed with cancer, at least in the initial stages or even at a late stage, when the patient is diagnosed, when we are receiving the blood sample, we will analyze the samples and then give a right decision to the doctor that this is the origin of cancer, for example, so that instead of giving a huge spectrum of chemotherapy or radiotherapy, they can personalize the treatment specific to the patient. That is personalized medicine to the patient. That is the overall idea. And then the patient can recover faster and go back, hopefully, to their healthy stage as they were before. That is the overall aim of the project. This is a schematic of the overall picture. What is the idea that the wood here is used. It's chemically processed in our collaborators' lab, and then we take the nanofibrils, the cellulose nanofibrils. We call it CNF. So those cellulose nanofibrils are modified in a specific way to bind antibodies to it, and then it captures the circulating tumor cells, that is CTC, in a blood flow. That is the overall idea. So how we achieve this? To start with, we have a silica glass, just a regular glass slide or a PDMS surface as a substrate. And then on top of it, we have different layers, starting from PAH, that is the stabilizing layer first that we coat on top of the silica glass or PDMS. It's polyallylamine hydrochloride, which is positively charged. And on top of that, we add a negatively charged layer, that is CNF, cellulose nanofibril, and then that is considered as one layer. And on top of that, we build polyethylene imine, which is a positive layer, and then we keep on repeating it until we reach at least 5 layers. So why do we need 5 layers in this case? Why not just 1 or 2 layers? I will answer that question shortly, which is very important as well. So after this layer is being built, we add the antibodies. In this case, it's anti-EpCAM. And then once it is captured and all the blood cells are being flushed out, we come up with a specific enzyme for digestion, and then we release the cellulose of interest that has the cancer cell attached to it. So, and then we can use these cancer cells for downstream analysis in the end. So how do we confirm that we have cellulose on top of the layer, on a glass or on a PDMS microfluidic chip? In this case, we used atomic force microscopy, where we can clearly see that one CNF layer all the way up to 5 CNF layer, that the layers are being built up. And then also from the graph, you can clearly see from layer 0, which is the baseline, layer 5 is completely being saturated, and we can see the peaks, how it is growing over different layers. So this is easy when it comes to doing it on a silicon chip, for example, and then we can scrape off the layer and then put it on a AFM machine and then look at it. But it is not practically possible to do that all the time to visualize the presence of cellulose. So that is when we stumbled upon how do we use it. So the initial approach was we used polyethylenimine in the previous slide, which I showed you, as a final layer, which was fluorescently labeled. But then the problem was that PEI was staining all the surface, irrespective of the presence of cellulose or not. So that gave us completely wrong results, that we were not staining just cellulose, but we were staining the entire surface, whatever was on its way, glass, PDMS, whatever. So that was not a practical solution for us. And that is when we found that we have this product called Carbotrace that can specifically stain the cellulose, and that was a magic at that time. I could not even believe that this was existing at that time. And we tested out, we reached out to the company, and then it was just right behind our office, and we didn't know that this existed. So we reached out to the company and asked for a test, and then we purchased the product and we tried this. And we can clearly see here that before and after release with 1 layer, and before and after release with 5 layers, the difference. That clearly only the CNF were very clearly being labeled here, and no background at all when it comes to Carbotrace. So it purely was binding to the cellulose in this case. So this was very important for us to characterize so many different parameters. What I'm showing here is just one parameter, but then obviously you might guess that this is not just one experiment of that we tried Carbotrace. We had used for different control experiments and validations of our process. And then once this is done, we were confident that the cellulose is present and then being released at the same time. And that's when we started moving to cell-based experiment. And here what you see is at 1 layer, the cells, the number what you see, 4,262, is basically the cells that is present before release on a 1 layer CNF. And then after enzyme digestion, the idea is to release all the cells. That is how it should work. But then because of 1 layer is not enough, because it does not cover a homogeneous layer on the glass slide or the PDMS, it was having a lot of unspecific binding of cancer cells to the PDMS or the glass slide. And that is when we increase the amount of layers we needed. That is, for example, in this case, 5 CNF layers. And you can clearly see a difference that spiking in 599 cells before release. And then after we release, we could almost release all of the cells from the surface. And that is why we needed 5 layers and not just 1 layer of CNF. So once this is done, we also wanted to show that how effective the washing is, or the enzyme digestion is. So we incubated the cellulose at different time intervals. So here you can see that starting from the 0 and 10, 20, 30 minutes. After 30 minutes, all the cellulose was completely washed away. And this wouldn't have been possible for us to analyze it if it was not Carbotrace. So it was really a key feature here for us to do these quick analysis. And then once we have a robust system to work, we moved on to a better chip that gives better mixing within the channel, which improves the probability of the cells getting attached to the antibodies, because it is usually when the cells flows in a microfluidic chip or the fluid flows in a microfluidic chip, it goes in a laminar flow. So how it enters will always exit the same way. But if it's going to happen like that, then we will miss out all the possible binding of CTCs onto the glass slide or the PDMS. So in that case, that is when we used the herringbone chip. What you see there is basically a lot of bumps, like speed breakers on our road, and then it basically creates a vortex within or mixing within the microfluidic chip, and that enhances the probability of cells adhering or getting attached to the antibodies that is being modified on the PDMS glass surface. What you see here is basically spiking in cancer cells at CT116 onto a blood sample and then flowing it through the chip, and then we washed it. There were very less specific binding of white blood cells, which is CD45, as you can see there, and there were a lot of CTCs were captured, or I should not say CTC, it's a cancer cell line. The cancer cells were captured that is CK18 positive, cytokeratin 18 positive. So this is a very good result for us to show as a proof of concept that the methodology worked overall. And then we also wanted to check, because we are going to release it, is it viable after being released? So after release, we collected and then we counted and we checked for the viability. And here you can see the viability was at least 97% of viability that we after being released. So that's very good for further downstream analysis for culturing it or doing any molecular characterization or anything like that. So once we have finished that, the idea is to move this platform to other platforms for downstream analysis. That is ongoing currently in my previous group back in KTH in Aman Rusom's lab. But this is one of the projects that I had during my PhD. To finally conclude, what was the end result of all this is, so using this technology of microfluidics, and then if you are able to get access to the circulating tumor cells, we can actually have so many different analyses being done on those single cell, look for progression studies, how the treatment has been working on the patient, or do cell culturing or grow organoids on it and then do drug testing on it. DNA, RNA analysis can be done, even protein analysis or sequencing can be done. So these are all the possibilities of having circulating tumor cells in the study. So here I thank and acknowledge my professor, Aman Russom, who gave me this opportunity to work, and then our clinical partner who supplied all our blood samples, all the blood samples for us to work on, Professor Matthias Lohr, and our cellulose partner who delivered all these genetic or not engineered CNF, that is Professor Torbjörn Pettersson, and then my colleagues Harisha and Ruben who worked with this project throughout the PhD study. So thank you for listening, and I'm happy to answer if you have any questions regarding that.